Okay students, let's recap all the main points of mitotic phase. We discussed the first phase, prophase, where nuclear membrane was prominent, thick. Then we discussed the formation of chromosomes after the process of phosphorylation, enzymatic activities. Chromatin material was changed into chromosomes. Centrioles, two centrioles moved to opposite poles. They gave rise to spindle fibers and esters and change into special apparatus that is called mitotic apparatus. Before entering into next phase, that is called metaphase, again some enzymatic activities are there which actually break down the laminin, the protein of the nuclear membrane. After phosphorylation, this membrane starts disappearing, and when we reach in the next phase, during metaphase, this nuclear membrane will disappear. Over there, a special structure is formed by the activities of spindle fibers. When they reach to opposite poles, they make a special structure that is called spindle. These chromosomes, which are actually a duplicated material, there are two sets of this genetic material. They were duplicated over here in S phase. Actually there was one set of chromosomes, 46 number of chromosomes were there. When two sets, one set was when duplicated, we had two sets, and total 92 number of chromosomes were there, two sets were there. The chromosomes actually arranged over here on the equator. They attach with the spindle fibers from a special position that is called kinetochore, which is present on centromere. Then these spindle fibers constrict, they pull away themselves towards the poles. They also pull up the chromosomes. When they pull up the chromosomes, chromatids will get divided and starts moving to two opposite poles. They are not half actually. We have two sets of chromosomes, 46 plus 46. So we have complete set of chromosomes on both sides. One set of chromosome will move to right and the second one will move to left. So up till now, the process of karyokinesis will be completed, during which the nucleus actually divides. After that, the second process starts, the cytokinesis. In cytokinesis, a cleavage is formed in cytoplasm or cell membrane, starts dividing. It starts moving inward, and finally it will get divided into two layers, and resultantly one parental cell will be divided into two daughter cells, and again we will be having the materials in the same state from where this process was started. Chromosomes uncoil, they again come back into chromatin materials. Thin nuclear membrane is there, which is not very prominent. So this is all about the process of mitosis. When we talk about the significance of mitosis, let's discuss some important features, the significance of mitosis. Importance. So when we talk about the importance of mitosis, number one, as we observed, mitosis is a process in which one parent cell divides into two daughter cells, and both these cells are genetically identical. This is the reason This process is carried out in all body cells. Wherever we have a need of new cells, if new cells are to be formed over there, definitely they should be genetically identical to their parental cells. When we get any cut on our skin, or when it is bruised, we have to replace the cells. In that case, when new cells are formed, they should match with the place from where they are removed. So that is the reason in this process we make genetically identical cells. So the first importance of this process is asexual reproduction. This process of mitosis occurs in some plants and animals for reproduction purpose, like bacteria, amoeba, yeast, and some other plants are there which use this process to produce their new generation. So that's why it's a part of asexual reproduction. Second, development and growth. When talk about the development and growth, when gametes fuse together during the process of fertilization, when zygote is formed, this zygote further will undergo in the state of mitosis. It will pass through series of mitosis, and during the process of development, it will be first divided into 2, then 4, then 8, and millions of cells will be formed. Definitely it will be changed into different layers, and those layers will give rise to different organs. So development and then even growth. A small seed will be changed into a taller plant. One zygotic cell will be changed into a new individual, which will be having a height of 5 to 6 feet. How it is possible? With the help of mitosis. This process continues, cell divides, redivides, new cells are formed. By the accumulation of those new cells, there is a process of development and growth. Cell replacement. Okay, when we talk about the cell replacement, as we discussed earlier, sometimes when we get cut on our fingers or any other part of the body, or when there is a bruise, skin is removed, during the process of replacement, when new cells are formed, when we get a new skin over that wounded area, so how do we get those cells? By the process of mitosis. If we get any cut and skin is removed, in that case the process of mitosis will get start, new cells will be formed, and in this way they are replaced. So cell replacement means the process of mitosis is also involved to replace the old worn out cells to make new cells over there. Then regeneration. We can take examples of different organisms. We can take the example of planaria. Then we can take the example of starfish. These are the animals, actually they do regeneration. Regeneration is a process in which the lost parts of the body can be regained. And when we talk about this planaria, it uses a process of fragmentation. When it will divide its body into different fragments, and with the help of mitosis, each fragment can be changed into a new organism. It's a regeneration process. Same is the case with starfish. If you cut any arm, it will replace, a new arm will be regenerated. Even you can take the example of lizard. If you remove the tail of the lizard, it will regenerate after some time. So which process will be involved there? The process of mitosis. So these are the significances, some benefits of mitosis.